Thanks, John, and for the kind words about the team at One Nucleus, but also I'd echo John's welcome to you here this morning. It's a pleasure to welcome you as Chief Exec of One Nucleus. I know you're here to listen to the content and the experts, not listen to me, so I'm not going to spend long before we move along to the main program. Obviously, we can only do these things because of the kind generosity of sponsors, the delegates turning up and people engaging, and people giving up their time to come and speak, give presentations, be on panels, and provoke those thoughts and questions, which ultimately is what moves innovation forward. So my gratitude to everyone that supports us for this event, but through the year, it's much appreciated. So the first session today, because we talked about links internationally, locally, innovation is driven around the world, I guess. And it goes because people work together and they challenge other ideas and they work in great teams. It's probably never been more interdisciplinary as a process, I don't think, not least because of the new modalities coming through that are complex therapeutics, how to discover them, develop them, regulate them, and actually how do you put the teams together that can do that. And I look up in the audience and there's people who are experts in building teams, there's experts in doing the science, and it's great to see how that gels into not just individual companies, but a sector that delivers so much good to the world. But without that, I think I'm going to welcome our first keynote speaker to set the scene for new modalities, Pedersen from Pelago Bio, who's run complex teams both sides of the Atlantic in big organizations, building those teams to be strategic and how you take on these major new challenges. So the bio's on the app, so we'll leave you go and sort of just see how stellar Kia is, but I'm looking forward to hearing your talk. Kia.
Thank you for that introduction, and thank you for giving me the opportunity to kick off this very interesting session on New Horizons for Therapeutic Modalities. And let me see which one. It's this big one. So we're about to embark on uncharted territory, but I think we know a couple of the obstacles that we're likely to encounter from our more mature modalities. So we know that when we're up against novel biology or a novel design of attacking a disease, there will be challenges. There will be challenges in delivering that therapy to the right organ, tissue, cell. And with that comes, of course, regulatory safety concerns. And none the least, manufacture new modalities. That has its own set of challenges, as has commercialization. So I actually figured I'm going to do a broad overview, shallow overview of the more mature modalities and what can we learn from them and how are we advancing the new modalities. But so we're in for uncharted territories, so you might wonder who am I to be your guide on this. So brief introduction to me. I have a PhD in cell and molecular biology. I wasn't born at AstraZeneca, but I was sure raised there. I learned how to do drug discovery the big pharma way, from target validation, designing high-throughput screening campaigns and your screening funnel, and bringing it all the way to candidate drug. I'd like to believe I took the best part of that into smaller companies, streamlining drug discovery. And then I switched gears, so I moved over to bioprocess instead. And again, I joined a really large company to learn how this is done. And I would lead R&D teams in developing instruments, consumables, processes, so that our customers could produce antibodies, gene therapies, vaccines in the most safe and effective way. And today, I'm Chief Operating Officer of Pelago Bioscience, and we're a contract research organization, so we support drug discovery throughout the value chain, understanding what is your target, how can we generate hits for it, and all the way to preclinical phase. using our patented method, CETSA. So this is not going to be a plug for CETSA, but me and Marianne are here, and we're more than interested in discussing with you how we can support your drug discovery journey. And we have knowledge from helping more than 200 customers through this, so please come talk to us. But back to the uncharted territory. And to look for new horizons, we need to know where are we currently. So current state, small molecules make up 90% of all drugs sold. There are more than 37,000 marketed products. If you slice it a different way, small molecules capture 60% of the market, so shrinking, but still very vital. And there are more than 14,000 programs in pipe based on small molecules. So we worked with small molecules for a long, long time, and we've learned a lot. And I think we all know the challenges of reaching the right target, and hopefully only the right target in the right patient, in the right tissue. So efficacy and safety are the major holdbacks of small molecules. And this is where, of course, we can learn for the new modalities. And I think this is also, 14,000 new programs. That is because we have a wealth of data to learn from that we can use to explore new modes of action. This is an area where for sure we can use AI and machine learning, because there's so much data to build on to progress small molecules faster. Another way of looking at where we are today is, of course, to look at what did we sell last year. And in clear lead are antibodies, led by the PD-1 blocker Keytruda, and followed by TNF-alpha blocker, TNF blocker Humira. And so if small molecules struggles with specificity sometimes, antibodies show exquisite specificity, and we've been using this for a long time, and purifying from animals, and then we overcome those hurdles that it entails when you inject animal material into humans by inventing the hybridoma technology and humanizing antibodies. So that was a big breakthrough for this modality. Not only would we avoid much of the immune response to the therapeutic antibody, but it was also more effective and had a stronger half-life. So this is a mature modality that we keep building on this one. And it wasn't hard to understand that, okay, we have this clear specificity on two arms of the antibody. What if we use this arm to attract T cells and this arm to attract to go for antigens on tumor cells? We can bring them together, and we can more selectively kill off tumor cells. So that bispecific antibodies, that was proposed already in 1960 or so. It took some time to overcome a lot of challenges. And again, one challenge is manufacturing, because now you have to produce an antibody with all the right specificities in all the right places. So knobs into holes methodology is just one way of addressing these hurdles. And much along the same lines, it isn't hard to envision how we can use the specificity of the antibody to bring a drug to those cells, so cytotoxic agents to kill off specifically tumor cells. So this is a good example of a modality that keeps evolving into new areas and where we build on the strengths of a more mature modality. Next on our top seller list is, of course, mRNA vaccines. I'm not going to talk about this because there are so many people who can speak to this success story and the details of it in a better way. And next one is, of course, course, Ozempic, another very successful story that I'm sure we haven't seen the last of. It's a very mature modality, recombinant proteins. There are more than 500 products on the market today for a lot of diseases. And again, we've used animals until we figured out how to recombinantly produce it, and then we could start modifying for different characteristics. Insulin being the most prominent example, of course. So this is paving the way for new exploration in that area. But this was last year. So what does the pipe look like? I said we have 14,000 small molecules programs in pipe, and then we have another 10,000 programs. And as you can see, a lot of them are directed towards monoclonal antibodies, bispecifics, ADCs, and followed by then recombinant peptides and proteins, which are having a revival of sorts. And then comes cell and gene therapy, CAR T-cell therapies. So let's talk a bit about cell and gene therapy, because 2023 was actually a breakthrough year for cell and gene therapy with 7 approvals by the FDA. So now we have around 40 cell and gene therapies approved and an additional 500 in pipeline. And so we're expecting that by 2025, we will have 10 to 20 new gene therapies every year. And you can do this in different ways. You can add the most often corrected genes into the human, and then you need to find a good delivery vehicle. Or you can extract cells, modify a gene in ex vivo, and then bring it back to the patient. And that, of course, comes with its own set of challenges. So CAR T-cell therapy is the most prominent ex vivo gene therapy, where we take T-cells from patients, we modify an antigen so that we direct them for, or a receptor so that we direct them for tumor cells. Again, challenges. We are using the immune system, and that is prone to backfire on us, so that's a major challenge with this modality per se. But then manufacturing is one of the big hurdles. This has to be done bedside. We have to take the cells, modify them, expand, enrich, and then bring it back to the patient. It's not only costly, it's tricky as well. So we're overcoming this by taking T cells from donors, healthy donors, and then modify it genetically. And this means we can simplify and standardize the process. So more consistent quality and lower cost, of course. And you can have the therapy at hand, so you can bring it to the patient immediately instead of having to go through the production cycle next to the patient. Again, of course, hurdles, and with this specifically, there's always the risk of rejection and graft versus host disease. Another way of doing gene therapy is to deliver straight into the patient, and then we have 2 options, the non-viral or the viral, and the viral pathway has proven more successful so far, and specifically adeno-associated viruses have been the choice for many. And adeno-associated viruses have a lot of pros. It can carry not a huge load of genetic material, unfortunately, but they come naturally with different serotypes, with different tissue tropism that we can explore and engineer further. There's also the benefits of being able to transduce non-dividing cells as well, and the no cytotoxicity and low immunogenicity. These were hurdles early on during gene therapy, that the immune system would go haywire with fatal outcome. And also, the low integration frequency is a clear benefit of AAV, because that was also one of the major hurdles early on. Of course, it has drawbacks. So the limited cargo capacity, as I said, and most of us have encountered AAVs, so we carry antibodies already. So that's what can make it less efficient. And then again, I'm going to come back to the manufacturing. Maybe it's because I've been working a lot with this, but for AAV, you need a lot of genetic or viral particles for each treatment. And there are a number of ways of producing these particles: plasmid post-transfection into mammalian cells, viral infection into mostly insect cells, and then, of course, the field is moving towards stable packaging and producer cell lines. But that is only the upstream part. Then we need to purify the material as well. And then there's the challenge of a lot of viral particles actually don't carry any genetic material or carry the wrong genetic material. So how do we get consistent batches of AAV particles? So with this very rapid run through, I hope I have set the stage on what we can learn, what obstacles we have overcome so far with the mature modalities, and how we can use them going forward for the advancing, emerging modalities and where we need to focus. So with that, I'm really interested to hear next presentations and discussions. Thank you.
Thank you, Kira. And Kira will be on the panel as we come to that part of it. So if we hang on to the questions that are burning away, we'll wait until then. Refreshment. So now, just illustrate that, I guess, before we get to the panel discussion to talk about some of those challenges and what's coming. We've got two of the hottest plays in new modality sector. So I'm delighted to have and asked to come to the podium, Amir from Resolution.
Good morning, everyone. My name is Amir Hefni. I'm the CEO of Resolution Therapeutics, and thank you to Helix for the invitation today. I'm going to build on Kia's presentation. She touched on the importance of cell therapy as a new modality, and we're trying to bring cell therapy to an area of great unmet need, which is end-stage liver disease, an area where so far many treatments have failed, and we hope we can turn this around through this novel approach. So at Resolution Therapeutics, our mission is to transform the outcomes of patients with inflammatory organ disease by pioneering macrophage cell therapy. As you're aware, macrophages present themselves in various phenotypes. Our focus is on pro-regenerative macrophages. Those are macrophages that exhibit anti-inflammatory and anti-fibrotic properties. Our objective here is to reduce the inflammation in the inflamed tissue, in the inflamed organ, resolve fibrosis, allowing the organ to regenerate and heal. So our first indication that we're going after, as I mentioned, is end-stage liver disease, and I'll talk a little bit more about that on the next slide. And what we've done at Resolution, building on some great work from the University of Edinburgh, so we're a spin-out from the University of Edinburgh, we've engineered the macrophages to allow them to deliver greater anti-inflammatory and anti-fibrotic properties, and in doing so, having not only a potent response in this patient population, but a durable response as well. And I'm going to touch on that bottom right quadrant, is a fundamental question. Does macrophage cell therapy work in in cirrhotic patients? And we have two studies, a phase 1 and a phase 2 study from the University of Edinburgh, actually very recently published at EASL, the European Association Study of Liver Disease in Milan, about 3 weeks ago, that answers that question. I'll touch on that data in a couple of slides' time. In terms of our team, we're UK-based biotech, so we're based between London and Edinburgh. As I mentioned, spun out from the University of Edinburgh, so we have a research organization up in Edinburgh, macrophage biology, and then our manufacturing, an autologous manufacturing cell therapy company, so our manufacturing organization is based in London. So in terms of the patient population we're going after, if I bring your attention to the left-hand side of the slide, as the liver becomes progressively more injured and patients move from a compensated cirrhotic state to a decompensated cirrhotic state, they're presenting in hospital with major adverse events or major clinical events such as ascites, variceal bleeding, hepatic encephalopathy, and that's where we're looking to administer our cell therapy RTX-001. For this patient population today, the only treatment option is a liver transplant. And about 30% of patients waiting for a liver transplant will pass away without getting a liver transplant. So a really, really high unmet need. If you look at the top right-hand chart, it shows mortality, so survival, and you can see there after a first decompensation, median survival is 2 years, a second decompensation is 9 months. So our objective at Resolution Therapeutics, or our vision, is not only to prevent major decompensations, but potentially to prevent the need for these patients to have a liver transplant. And we've plotted on there in the dotted line the other CAR-T or other indications where cell therapy has been approved, and it's had transformative impact on these patients. So diffuse large B-cell lymphoma and multiple myeloma, where a significant portion of those patients have been cured with CAR-T cell therapy. And on the bottom right, even with the introduction of GLPs and other forms of earlier therapy and earlier lines of treatment, this patient population unfortunately continues to grow into the future. I touched on this already. We're a spin-out from the University of Edinburgh, largely driven by the work from Professor Stuart Forbes, who's a professor of regenerative medicine at the University of Edinburgh, and has studied macrophage biology for about 15 years in a preclinical setting, in vitro and in vivo, and more recently launched 2 clinical studies, a phase 1 and phase 2, in partnership with John Campbell, again from the University of Edinburgh, and I'll touch on the data in a couple of minutes. And what we've done, as I mentioned, we've engineered with 2 payloads. This is mRNA engineering into the cytoplasm, so we're not using viral vectors. We've specifically gone after an mRNA strategy into the cytoplasm, transient engineering that allows the macrophage to express 2 genes and deliver a cytokine and an antifibrotic agent that exists within the natural microenvironment of the macrophage, and in doing so, delivering additional anti-inflammatory and antifibrotic properties. Why is this important? How do macrophages exhibit the effects that you'll see when I walk you through the clinical studies? It's really around 2 key axes. Again, a reminder that our focus is pro-regenerative macrophages. We're not going after pro-inflammatory macrophages. In Kia's presentation, she had CAR-M as one of the new modalities. So we know out there's a number of companies that are pursuing pro-inflammatory macrophages to try to change and impact the tumor microenvironment. In our case, we're going after what was traditionally called the M2 phenotype, so anti-inflammatory, antifibrotic, to reduce inflammation and address fibrosis. And doing it via the mechanism of action that you see on the chart there, anti-inflammatory, phagocytosis, bringing in monocytes from the innate immune system, being able to differentiate them into pro-regenerative macrophages to continue the work that the infused macrophages have started. And then from an anti-inflammatory perspective, not only breaking down the fibrotic scar tissue, removing the extracellular matrix, but also preventing new scar formation as well. If I translate this slide into data, and this is a really busy slide, but just a very high level. On the left-hand side, it's in vitro data. And what you can see when we've plotted non-engineered versus engineered is a significant improvement in the ability of the engineered macrophages to prevent scar formation, to bring in monocytes from circulation and polarize them towards a pro-regenerative phenotype. And to us, that's really critical from a durability of response perspective. And then on the left-hand side or right-hand side, your right-hand side of the slide, this is part of our GLP tox that we submitted to the MHRA. And you can see a couple of things there, relatively well tolerated. So we don't see the CRSs, the ICANS that you see with CAR-T therapy. You don't see the cytotoxic effect that you see with T-cell therapy. And then on the far right, you'll see there, reported by an independent histopathologist, this is blinded data, that we saw significant regression of fibrosis in a CCL4 fibrotic mouse model. So what we're seeing in vitro and in vivo is RTX-001, the engineered macrophage, being able to deliver that antifibrotic and anti-inflammatory effect. So why do we believe that we're onto a really exciting modality? I'll spend a couple of minutes on this. So I'm going to go back to the University of Edinburgh. As I mentioned, they conducted 2 clinical studies, a phase 1 and a phase 2. The phase 1 is published in Nature Medicine on the left-hand side of the slide. They show no major adverse events observed that you would expect to see in the CAR-T space. Furthermore, these patients don't require any lymphodepletion, no preconditioning, and no bridging therapy. And then the the signal of efficacy and the well-tolerated results led to them carrying out a phase 2 study in Edinburgh, 3 sites in Scotland, where they looked at a much bigger patient population with efficacy being an endpoint. And you can see there are 50 patients in the total in the study, 26 treated and 24 in the control arm. And in the interest of time, I'll move to the results. So on the left-hand side, you can see safety. Just at a very high level, this is well tolerated. I spent the last 4 years at Novartis working, leading the cell and gene therapy organization oncology there. And clearly, if we'd seen this with CAR-T, we'd be very happy. It's relatively well tolerated, no major cytotoxic effects. And what was really interesting is what we see from an efficacy perspective. We see 2 major events, 2 deaths in the treatment arm versus 10 major clinical events in the control arm. It was initially a 12-month study, extended for 18 months follow-up. So this is the 2.5-year data. We now have 3-year data that shows even greater response rates. If we translate this to Kaplan-Meier, as we're used to in oncology in terms of looking at survival, you can see there is significant differentiation or opening of the curves between treatment and control. And you can see there that the results seem to be durable with non-engineered. Our objective is Resolution Therapeutic. We're going into a slightly sicker patient population. I would say this is primarily a cirrhotic patient population in the MATCH study, with some patients that have had prior decompensation. Where we're going into are patients that have had their first decompensation, 6 weeks to stabilize, and are infused. So we're going into a sicker patient population, hence why we believe the engineering will have a greater impact. What I'll do is I'll just touch on this slide and then open up for any questions that you may have. So where are we in our journey? We have our own natural history study that's taking place in the UK and Spain. Then we just received CTA approval from the MHRA last week, so we're very excited as a small biotech to have the go-ahead for our clinical study. We have first patient first visit in September this year, and we look for primary analysis in December 2026. So with that, I'll stop and just see if you have any questions, and thank you again for the opportunity to present Resolution Therapeutics.
Thank you, Amir. If we keep the questions for the panel, it won't preempt the discussion at that point. So it's now a pleasure to call up Madeleine from Harness. Another hot topic coming through. Over to you.
Thank you. Good morning, everyone, and thank you very much for the opportunity to come and tell you a little bit about Team Harness, who you can see there. So we are 22 people. We're based at this campus over in the Minerva building. And the team is mainly based in research with deep experience in RNA biology, neuroscience, and bioinformatics. The company was founded in 2020. And some of you may remember us as being Transline before we were Harness. And be aware that, like many biotech companies, we've gone through a process of pivoting and rebranding the company. And that's mainly because the tech that we were founded on, so synapse, which are long non-coding RNAs that interact with the ribosome in order to increase the rate of translation, is a fantastic concept. But after we've been working on them for a couple of years, we realized that they need a little bit more time to optimize before we take them forward as a therapeutic. So the decision was taken to broaden the company's toolbox, to focus down on neurodegeneration, but with that very same mission of upregulating in a controlled way. So let me tell you first of all about controlled upregulation. So why controlled upregulation? So traditionally, as we all know, the industry is really focused on knocking things down rather than knocking things up. And that's really because it's easier to knock down expression of a deleterious gene or a protein than it is to increase things just by a little bit. You might argue that gene therapy does that, and it does that very well. But often that results in uncontrolled expression, where you're getting 10x or 100x fold expression, and you can't turn it off. And that's not suitable for many targets, particularly in the CNS, particularly the target I'm about to tell you about now, which is the one we're working on for our lead program, FAN1 nuclease. So that's heavily expressed in the brain. You can understand why you wouldn't want to necessarily flood the brain with a nuclease. So what we're trying to do is harness the body's endogenous systems, post-transcriptionally and translationally, in order to bring about this small effect. And that's where the name comes in, harness. So easier in some ways to understand than transline. Our ethos at Harness and what we're trying to do is to focus on a deep understanding of that target biology in order to choose the best upregulation strategy for a particular target. So we have a variety of modalities that we're trying to do that with and a holistic approach to modulating translation. So once we've identified the target gene of interest, we're looking for hotspots in that transcript in order to work with, and they might be in the 5' or the 3' UTR in the coding region itself. In the 5' UTR, it might be something like an upstream open reading frame, which is reducing the efficiency of the proper open reading frame, so we might be able to damp that down or turn that off. It might be a splice modulation approach in the coding region, or in the 3' UTR, it might be something like identifying a microRNA binding site that we can modulate there. So we use a variety of different modalities, steric blocking ASOs, base editing approaches, and then in the future, our sign-up technology will come back into the fore in its next generation as well. So one of the challenges that we've faced, and one of the things that we've been investing in quite heavily over the last couple of years, is obviously quite hard when you're only making very small changes to proteins to have assays that can measure those reliably. And it's not something that you can really do with a Western blot, which is often semi-qualitative at best, or semi-quantitative at best. So we've invested quite heavily in generating engineered cell lines. We take a high-bit tag and we engineer them on the C-terminus of our target genes of interest. And then we can read out with a luminescent signal and really cut back on the noise. And obviously, immortalized cell lines are really great workhorses for screening, but actually there's no substitute for being in the target cell. So another thing that we've been investing in very heavily is bringing in iPSC and neuronal skill sets in-house so that we can generate complex functional assays that give us a much more clinically representative readout for in vitro efficacy. So let me change tack a little bit and give you a real example of what we're trying to do with our novel approach. And a really horrible, terrible neurodegenerative disease called Huntington's disease. So when I was at university, the Huntington gene had just been discovered, so that was back in 1993, which tells you how old I am. And it was, we were taught that HD was caused by triplet expansion in the HD gene. And it led to the expression of a protein, mutant Huntington, with polyglutamine tracks that then stuck together and aggregated, and that's what was causing the neuronal dysfunction and death. And what's been happening in the last few years is a real revolution in the thinking in the field now. The disease is not really, or the primary pathophysiology of the disease is not this aggregation of the Huntington protein. That seems to happen right at the end of the neuron's life cycle. And what's causing the transcriptional dysregulation and translational dysregulation that's upsetting the neuron and causing this dysfunction is in fact the CAG repeats themselves. So it's a function of the DNA and the RNA as much as it is of the protein. So firstly, let me just tell you a little bit about that. So very briefly, people are born with a certain number of repeats in the Huntington gene. If you've got 40 or more, then you will definitely get Huntington's at some time in your life, unless something else interrupts first. And as you age, the number of repeats in these neurons increases over time by a process called somatic instability. And they're not happening in a synchronous way, it's happening in a stochastic way. So it's only when enough neurons have been made dysfunctional and die that you're starting to see symptoms of the disease, although biomarker evidence of the disease is apparent much earlier. And this process is being driven, this process of somatic expansion is being driven by the DNA repair mechanism. So you've got can't get the laser to work. A complex called mutase beta, which is involved in mismatch repair. And when you've got a long section of DNA with repeats in it, you get slips and loopouts, and it comes along and it fixes these loopouts of DNA. What it does in the process is it incorporates new flaps and actually makes the problem worse. Then there's an enzyme called FAN1, a nuclease, that comes along and it excises the flaps and fixes the problem. So you can see that they're acting in kind of a seesaw fashion. So if we can elevate FAN1 and elevate the seesaw that way, we can keep a lid on what mutase beta is doing and slow down or possibly even halt this process of somatic instability. So when you get HD, isn't just governed by the number of repeats that you're born with, it's also based on a number of genetic modifiers, and these have been identified using genome-wide association studies. The one that we're working on, FAN1, is the one with the strongest association in HD. And SNPs, single nucleotide polymorphisms, in the FAN1 gene are associated with either accelerated onset of the disease or delayed onset of the disease. So it's a really good one that we can modify. And how can we do that? So this is one of the examples that we're working on at the moment. It's probably our lead strategy, but we've got others. So looking at the 3' UTR of the transcript, we find that the SNPs that were identified in the GWAS studies actually overlapped with microRNA seed sites in 3' UTR. And that's what's getting this delay of onset or acceleration of onset in the disease. So we thought, how can we interfere with that? And then using steric blocking ASOs to the seed sites in the 3' UTR, we can then interfere and stop the transcript from being targeted into the RISC complex. Hence, more transcript, elevation of protein expression, which we can then measure in our engineered cell lines, and we get this lovely high-bit signal, which you can see. This is work of in our lab, and at the moment we're getting around 30% elevation. This is currently being optimized both in terms of the sequence and the chemistry of the ASOs, but we know from the genetic information that it's probably in the right ballpark for what we need anyway. So meanwhile, we're also developing iPSC neuronal assays that are helping us. So you can get iPSC lines that come from patients that have got CAGs in them, long CAG repeats, and these can be expanded in vitro. So we've got a tool there that we can add our ASOs into in order to suppress CAG expansion in these neuronal cells. So with that, I'll probably wrap up because I can see that the clock is coming towards the end of the time, and Tony's actually getting up again, so that's my signal to wrap it up. So I just wanted to leave you with, it's obviously a very brief snapshot and a lot of complex biology to explain in 10 minutes, but also to say that we're very excited about our next program that's coming through the pipeline. It's related to nuclear import-export and is involved in TDP43 pathology. So we want to take that one forward for ALS, but it's obviously important in a number of other CNS indications as well. So with that, thank you very much for your time and attention, and looking forward to the panel discussion.
Thank you, Madeleine. So I think we saw in the overview some of the challenges there. We've seen some great examples of innovation, but some of the challenges still to face. And so I'm delighted to ask Hugh Nuthall from Eli Lilly to come forward and moderate the panel with the speakers and guests to explore those points a bit further. So Hugh, over to you, and panelists, if you'd like to come up, that'd be great.
Tony. So we still, what's the kind of time aiming? We still to 20 past still?
I know.
Yeah. Okay. Right, so yeah, thank you everyone for coming today. Thanks for joining us for this discussion. Thank you to our panelists for agreeing to take part. Briefly then, we'll just introduce ourselves. So my name's Hugh Nuthall. I'm from Eli Lilly, where I work in our external innovation team. Specifically, I'm interested in neuroscience, but I work with colleagues across all our different therapeutic areas of interest. And mainly, I'm part of the scientific and technical part of business development. So our role is to evaluate opportunities out there in all of your great companies, and where we're aiming to try and in-license assets, programs, and technologies, which will then go on and work together with you to discover and develop the next generation of therapeutics. So that's what I do. Over to our panelists. Amir, you've already introduced yourself.
Hi, I'm Madeleine Wakeling from Therapeutics. I'm SVP Program Management and Operations for the company. My background, originally a molecular biologist and a virologist. I've spent the last 20-odd years now in biotech and pharma and academia, developing drugs basically through from research into early clinical development.
Hi everybody, I'm Ben Taylor from AstraZeneca. I'm part of the Discovery Sciences department, which is involved in early phase drug discovery And I lead a team that's focused on gene engineering and mainly involved in gene engineering of CAR T cells to try and find ways to improve manufacturing processes and using gene engineering to find ways to enhance and boost CAR T cell potency to support our oncology and autoimmune programs.
My name is Davide Lucrezia. I'm the CEO and founder of Officinae Bio. We help customer and cell and gene therapy companies to design safer and more potent mRNA, leveraging machine learning to engineer non-coding region to ensure that the target is expressed in the right tissue at the right time.
And I think I did my presentation as well, so.
Yeah, we know who you are, Kia. That's great. So thank you, everyone, for introducing yourselves. So, yeah, I think we have a broad range of interests and approaches to therapies. So I think that's what we wanted to just kick things off with, some thoughts maybe around the terminologies. You know, how do we actually view a therapeutic modality? I think we all have probably different opinions. So maybe, Kia, I know you gave us a great overview broadly, but any one or two thoughts on how you would define that?
No, but I think there are many ways to slice it, right? It's about the biology or the way of delivering. So there are many ways to slice what is a new modality. And as we see, they keep evolving and building on each other. So it's not even hard. It's not even easy to say, is this a new modality or just an evolvement of an old one? So I don't have a sharp answer for that one. I don't know what the rest of you is like.
David, any thoughts from your perspective?
I totally agree that there is a different way to slice it. I would just like to see also added as a modality the dimension of the final impact on the patient. Some modalities do not change permanently the patient genome, others do. So I think this should be factored in as we move toward more sophisticated modality to really factor in the long-term impact on the patient.
Great, yeah, good additional perspective. Ben, any?
I mean, I agree. It's a lot of different things from mode of action to delivery. And I mean, also agree that it is using old technologies in new ways as well, right? And a lot of it is sort of changing the kind of brute force way we've been approaching disease to, you know, just basically inhibit something or block something to a more kind of nuanced approach to, you know, crafting things and, you know, subtle changes like we're seeing with the ASO approaches.
Mm-hmm. And I think sometimes when it's a new modality or using a modality in a new way and the target biology, or in fact the biology and the rules of engagement aren't fully understood, then there's some work to do there in order to catch up, not just to help industry, but to help the regulators then understand how we deal with these things.
I feel like my fellow panelists have covered all. I guess you know. I mean, you know, Ben's role, for example, looking at how do you take CAR T to the next level. I think, although we're autologous macrophage cell therapy company, we're leveraging a lot of the expertise from CAR T to learn, to evolve, and make sure that we take that on board as we get to clinic. So I think, you know, the learnings are really critical.
Yeah. So no one definition, but I think we kind of know it when you see it, maybe what a modality is. But anyway, thank you for those initial thoughts. And that tees up, I think, our first kind of discussion topic, which is around kind of regulatory and commercial challenges for some of these new modality, new approaches. There's clearly been a well-trodden path for some of the more established approaches, small molecules, antibodies, peptides over many decades. But I think some of these new approaches need a new way of thinking. So maybe Madeleine, I was going to ask you to kick things off from a nucleic acid perspective. Any thoughts there on regulatory or commercial?
Yeah, so obviously we're working on oligonucleotide products, and they're not brand new, so that they're not the babies of the field, but they're definitely in their teenage years, I think, with only 17 currently approved therapies. And oligos are a funny one with the regulators, aren't they? Because they sort of fall between the two stools of small molecules and biologics. So often we're developing them in a way that's suitable for biologics, and then say if you end up going to the FDA, you're going to CDER rather than CBER and having a small molecule approach imposed upon you. So I think the regulators are still sort of catching up and learning. It's about understanding what the rules of engagement are and bringing the data forward and having that conversation. And there's some new guidance coming out. So there's a new ICH working group on oligonucleotides that was endorsed last year. Not heard much come out yet, so that's going a little bit slowly perhaps. And there's some new FDA guidance on non-clinical pharmacology that came out very recently as well. So it's emerging. But I think the key thing, and as we all know as developers as well, it's early engagement, isn't it? Getting in early, having the conversation, and developing in partnership.
And I guess you can have hybrid situations where you maybe got an ASO conjugated to something else. Does that change that conversation, or is it?
Well, that's even more novel. Yeah, absolutely.
So maybe from the cell perspective, I don't know, Ben or Amir, if you want to have any thoughts on the regulatory framework for those currently and how that's developing.
I mean, I guess autologous is quite a known and understood field. I mean, allogeneic is a lot more complicated, especially as we get into the more heavily gene engineered and modified allo therapies. And there, how are the regulators going to view all the gene modifications we're going to do? What kind of safety packages do we need? There's still a lot to understand there, and a lot of, and again, I think it's like you said, early engagement, trying to get those answers early, trying to cover as much ground as possible.
Yeah.
Just reflecting on some recent interactions, I think I feel like the MHRA in the UK is really open for early dialogue, and we've had some really good partnership, almost in some situations, co-creation of how we should approach, whether it's preclinical, critically the CMC package. So really, really pleased to see that sort of collaboration with the MHRA, and I think the FDA is advocating for the same thing. So we've had recently a Type C engagement with the FDA, which has gone really positive in a completely new modality therapy area. So I think the earlier you can have those discussions, the better. And I think maybe, Hugh, to your second part of the question around commercialization, I cannot highlight how important it is to think about things like COGS, vein-to-vein time, the kind of key manufacturing components of autologous cell therapy as early as possible. At the end of the day, when you commercialize, it's got to make commercial sense. It's not only going to drive revenue, but profitability for companies, and I think that's a big challenge right now for the cell and gene space, and we see that reflected in investor sentiment. So I think, as early as possible, thinking through CMC is really, really critical.
Which I guess is the case for all therapeutic approaches, but maybe even more so in this instance.
Given how expensive and resource-intensive autologous cell therapy is.
So maybe that's a good place to pivot to one of our next kind of discussion topics, which is really a kind of general one, which is all about, our collective goal is to bring forward novel therapies as quickly and as safely as possible. So really it was more of a round the panel discussion about, how are you individually in your organizations thinking about doing that specifically to bring things forward innovatively and creatively? Maybe we'll just go across Amir.
I'll go back to the conversation that we had earlier. I think the early engagement piece is really, really critical with regulatory authorities. I think also one of the things that we're trying to do is work with very close, so our partners, so our mRNA supplier, for example, working in partnership versus it's no longer a transactional relationship. It truly is a partnership building on where we are today for our phase 1/2 study, but what do we need for our phase 3 study? What do we need for commercialization? What do we need to leapfrog autologous into potentially other modalities? So I think that early engagement with regulatory authorities, but equally so with key suppliers, is really important.
I think where we have the win in ASOs compared to the cell therapies is possibly on the CMC.
Right.
Just sitting here feeling a little bit smug that having spent most of my career developing viral products or antibodies and other biologics, actually CMC for chemically synthesized things is quite rapid and easy. I was saying that, touch wood. But that's one of the ways that we can accelerate. I agree with you totally on the early engagement piece. And being, I think, prepared to think creatively or suggest creative things to the regulators as well in terms of your preclinical. And for us, it's thinking, because we're in neurodegenerative disease and CNS, thinking about the endpoints that we select for the clinic and trying to find things that show an earlier signal, because obviously neuro is very long-winded. So yeah, that's how we can do it.
So you think that the ASOs and other oligotherapies can be more quick in the development path than some of these more traditional approaches, or?
I'm not sure about that, but I think being very used to CMC being on the critical path,
Right.
it's nice to have something else being on the
Right. Ben, yeah.
I mean, I guess thinking about ASOs, you know, what we've seen from our internal programs is compared to traditional small molecule discovery, ASOs are really fast.
Yeah.
So that's definitely one way of getting to the clinic quicker, choosing the right modality that will work for that particular disease, and choosing the fastest one, you can progress as well. And then in terms of cell therapy, I mean, I think what we need there is a lot of development in manufacturing processes. And it's not really so much, especially in the autologous, it's not really so much the actual manufacturing run of the cell itself. It's really the lot release processes that are the major bottleneck. And how we change that and get kind of in-process testing to be able to get those materials released and into the clinics much sooner is going to be the next big challenge we face. And then for the allo side, how we can simply reduce the manufacturing times. There, lot release isn't really a problem. It's you know, we want to do a complex manufacturing process, maybe long expansion runs. How can we shrink that to a short and reproducible process as possible?
Referring more to what we are seeing with our customer, I think that would be very convenient to focus on safety as early as possible, because surprisingly, the highest friction point is not in the discovery, is when you go in preclinical. And that's where really the company has committed a lot. So any failure there is very impactful. And so working creatively could be really to join forces with who designed the payload and the delivery system to address the therapeutic windows as early as possible. For instance, we usually tend to work with companies that design the delivery system in order that in combination with our UTRs, non-coding region, you can really target the right cell at the right time and therefore increasing the therapeutic windows. I think this will really impact the friction rate of product through the pipeline.
Yeah, I couldn't agree more. Coming from the supplier side, this is where the fun starts. I mean, at GE, we would cater to production of antibodies. And even if we came up with a more effective, safer way of production, no one would want to change because everything was established, platforms were there were no savings to be done. But working with gene therapy and co-creating, that is so much fun, because then you bring all the different disciplines together and you sit with electrical engineers and software engineers to make manufacturing the most safe and cost-effective. So that's where we've had the most fun and being able to really make a change. So, yeah. Being invited to the table early really makes a difference.
Also because, if I may say, the later you start optimizing and risk management, the harder it is, because the closer you get to the filing, the less room for improvement and changing you have. So you have really to make an action as early as possible in order to minimize this kind of friction. Because when you get at the end of your preclinical study, perhaps it's too costly, perhaps too late to really make a pivot.
So a few of you touched there, I think, on kind of safety aspects of some of these newer approaches. Are there any key messages there around particular safety concepts or thoughts for some of these newer approaches versus the more well-trodden small molecule antibody approaches?
I think for us, ASOs have a reputation, so when they're given systemically, they're very given to liver and kidney tox. So that's not been our approach. We want to go straight into the CNS compartment. So one of the things that we've been hoping for, is that BBB crossing technology will emerge and evolve more quickly and achieve clinical precedence, because that would obviously be very helpful for us, but it's not there yet.
Right.
So we're going in with a CNS device straight into the brain. But I'm trying to do that intracerebroventricularly rather than intrathecally, because then you can go in with a much lower dose And the issue sometimes with ASOs is these narrow therapeutic windows. So very conscious of the safety and using that to bring to our delivery approach from the start.
And some kind of associated problems with the spinal cord tox, etc.
Lumbar scarring, particularly with products like tominersen.
Right. Anything else? I think that there's also the thought that you could, I mean, you touched on it, Ben, about being quite patient-centric, so maybe there's ways to go into patients earlier. I know you do that a lot in oncology and some other diseases anyway, but is that something that you think is more, that will become more of the typical approach for some of these newer cell treatments?
And I think that's where we'll see really big benefits particularly with cell therapies, going into earlier lines of treatment, instead of, as is typical now, going in third, fourth line of treatment. Can we get these first line? With these sorts of therapies, especially autologous, where you're manufacturing from patient cells, you need the patient cells to be as healthy as possible. If you're going in very late and the patient's very, very sick, it's hard to manufacture, and there are lots of manufacture failures. So you go in earlier, you can have better outcomes, better products, and potentially curative treatments, and that can really offset some of the costs as well and make the cost benefit a lot better. So I think pushing it to as early line as possible is going to be good. Difficulty there is the cost, right? And health providers are going to go with more traditional, cheaper approaches first before they invest in something very heavy. So how can we change that mentality? It's a difficult question.
And so that begs the question about cheaper alternatives, and is there still a space? You said here for small molecules. I mean, they're up there still as a large percentage of the number of treatments out there already. Is there still a space for small molecules in the future, or do you think that all these new clever approaches will supersede that?
No, I mean, we're building our toolbox, right? And then it's about using the right tool to address the right disease. So there's definitely a space for small molecules to evolve further with all the benefits that come with small molecules, and then you don't use them for the others where there are more sensible tools to use. And again, we were talking about safety. Our method, if I can plug some of our method now, that is about de-risking early. So I think the more we learn about small molecules and the different modalities, the more we can predict in vitro, right? So we can take the hit early on. That's what we're all striving for. But again, it means we need to sit down the different disciplines and understand how can we do this more, how can we do this smarter based on everything we know. And like I said, AI, ML, you need a lot of good data to be able to use it. And I would say that small molecules have an advantage there because there is a wealth of data. So then it's about collaborating and releasing that data so that we can use it together as a life science field. But I have hopes for that.
Good. Mm-hmm. Anyone else have strong thoughts? Do they see the death knell for small molecules, or still a robust future for where my background is? So I hope there is. Because
I don't think it's either or, right? Like it's in combination. I mean, I think I think we can talk about bringing cell therapy to first line for a number of oncology and non-oncology indications. But if there's a very good treatment, let's take diffuse large B-cell lymphoma. We know R-CHOP has a 60% curative rate. So actually, the first line setting is relatively well addressed. Of course, we want to increase that response rate, but maybe cell therapy is better suited for a second line in this particular disease area. So I think it's marrying up the unmet needs, the side effect profile, the cost of the health economy, with the modality that you're trying to introduce. So I think it's, we've got to, I think patients should have options, absolutely. Yeah.
I think just to add on that, I mean, it's good to have options, but for something like a cell therapy, lots of people cannot access it. You have to be in specialist centers, and lots of people cannot access it and cannot afford it. So, you're always going to have a lot of other options, and a small molecule just provides that patient accessibility that other treatments don't. So I think there's still a lot of space for small molecules, and especially with the the new approaches to small molecules like the PROTACs, the glues, the stabilizers, and all of those sorts of things, there's a lot more we can do with small molecules than we're currently doing.
Yep. Good. So maybe just to kind of round things up then, so we can leave a bit of time for questions. You touched on it actually, Kia. You mentioned kind of where AI and ML, machine learning, may touch on small molecules, but yeah, wondered if everyone could give a thought on how they are currently using these new approaches of artificial intelligence, machine learning currently, or where they think it will have a big impact in the future.
Yeah, so small molecules is an obvious one, but then again, production is another one where we can benefit enormously from AI help in making it more effective and safer. And I mean, we started with the digital twins and so on so that we can make manufacturing safer and more effective, but there's untapped potential in that end as well. So,
Like optimizing the lipid nanoparticle composition. That's an high-density data problem, so it's perfectly suited for machine learning. Choosing UTRs or selectively design antibody and proteins. I think that the cost per data point in biology is dropping down really dramatically, so it means that we have access to a vast amount of data over huge dimensional space, so we need machine learning to make sense of this. But certainly using data is super important. How to capture and standardize how we produce data is as important as about consuming it. So I would also focus on really harmonizing the procedure to produce data before consuming it. Otherwise, machine learning can help.
Yeah, and I mean, we use these sorts of machine learning approaches in pretty much every aspect of drug discovery now. And I think that is the key point, right? We sit on massive amounts of historical data, but a lot of it's totally useless. Because, you know, it's sitting in silos that are just no good to anybody. So how can we access those? Can we, you know, find better ways to pull that data out of the data swamps and data lakes and use it better?
Yeah.
I mean, if we can do that.
Metadata to make sense of totally agree.
I think from our perspective, using AI from a bioinformatics perspective to crunch huge amounts of publicly available data sets and then data that we generate in our own labs to identify those little regions in the genome would be of enormous value. And I'm not a chemist, but it seems to me that looking at chemistry of ASOs where you put the different modifications would benefit hugely from a machine learning approach as well.
So I've had a really positive experience and a not so great experience. I think we've seen it work. We've seen machine learning work really well from a manufacturing perspective, detecting particulates, for example, and going back to the root cause analysis and understanding why that's the case. But we've also opened up our database in a previous company to a very well-known major AI firm that promised to be able to more effectively analyze the data than we had internally, and I think the outputs were rather disappointing. So I think that for sure, you know, great data leads to great results, but if you have poor data and then you have, you know, you put it into a black box, then the output's going to be poor as well, and I think that data piece is so critical. So it made us realize internally that maybe we should have better data. Right. I'm optimistic, but I don't think we're quite there yet.
Not a magic wand, but we can use it in the best way. Right, well, I think that's a good place to end. Maybe before we go to Q&A, we can thank our panelists for a great discussion. And
Hi, Mark Eccleston. I'm representing a company called Volition today, but I do other things. So my question is related to safety. So in the more advanced cellular therapy type approaches, do you think there's an opportunity to integrate a different approach to safety? So rather than going from animal tox, which is not necessarily representative of the immune response, and then straight into humans for phase 1, you've got opportunities there for more sophisticated ex vivo tox, even on an individualized patient basis, because you're going to spend time manufacturing these medicines, right, on a per patient basis. You can take skin biopsies, things like that. There's things you can do with those patients directly to predict potential adverse events and maybe pre-treat the patients back to small molecules to downregulate adverse events before you then give the advanced therapies.
Anyone want to take that challenging question?
So, yeah, it is a challenging question. Yeah, I mean, yes is my answer. So we're doing a lot to try and build our non-animal model suite. So we have a lot of work ongoing with kind of organ-on-chip models using human-derived organoids and iPS-derived organoids to try and build better preclinical models to use both in discovery and in safety. There's a lot of work ongoing. Has any of it actually been used for any filing as yet? No. It's still all using the traditional approaches, and I think there's still a huge amount of work to understand and build kind of the confidence in those models that will then be used in filings, and how we can kind of bring the community together to build that confidence is going to be a big challenge, I think. And there are a lot of consortiums looking at this sort of thing and trying to build this community. So I think that's one thing. Going from patient to patient and using patients to actually pre-screen and test, I think is a great idea. A lot of challenges logistically and rolling that out, but I think that would be a fascinating approach, especially with quite complicated therapies like cell and gene therapies. Yeah.
I think maybe just adding to Ben, I mean, if you look at the recent approvals in CAR T space, I'm unaware of any primate studies, for example. Primarily cell lines, some rodent models, but relatively small versus what was traditionally expected from health authorities. So I think directionally we're heading in that direction. I've seen us use, in a previous company, we've used some simulation data to support some in vivo data and complement versus take over. But I think directionally there's an appetite by regulatory authorities to do things differently. Yeah. I don't think we're heading in that direction. We're maybe not fully there yet.
There's first approval, right, of a drug that wasn't put through animals. So it's basically, yeah, like you say, the regulators are willing to accept that if you convince them that what you're doing is the right thing. Yeah.
Actually, in fact, like there's an entire portion of a dossier that we had was based on purely modeling data, and it was received relatively well by the FDA.
Madeleine, I think you wanted to say.
I was just going to say, I think particularly the MHRA will give you pushback on doing what they would see as unnecessary cyno studies, and they want you to be able to justify based on your science what is the best rationale forward in order to prove that safety. And if you've got a truly human-specific product. And we will probably as well, because the things that we're playing with in the 3' or the 5' UTR of the transcript may not necessarily be conserved across species. Right. So you have to be able to go with the thing that makes the most sense.
I mean, in cell therapy, you're using human cells in a mouse model. That in itself is questionable whether it reflects what you'll see in humans. So.
That's it, or you end up with some kind of convoluted humanized model or a mouse surrogate that doesn't really compare to your actual drug. So.
Hi there, Matthew Fife, Storm Therapeutics. Benjamin, you said that antisense development was much, much more rapid than small molecules. Can you outline why that is? And then also, can you just give an idea about comparing the developability times of each of the modalities?
Okay, so, I mean, our traditional small molecule approach is screen the compound collection, 2 million compounds. That takes a good chunk of time to build all of those assays. So, it's 6 months, 1 year before you even do the screen. And then that deconvolution time and optimization, chemistry, iterative chemistry can take 2, 3, 4, 5 years until you've got a lead candidate that you can then take forward. In ASO, you've got a small window, you've got a limited number of sequences you can go after, you've got a wide chemistry space you can explore. I mean, probably better if if you explain this, right, rather than me.
I think it's probably better if Matthew explains it, because this is very much his area. But yeah, it depends whether you're going to tile across and go, looking at thousands and thousands of different sequences, or if you know where the sequence is and then you're iterating on the chemistry. But yeah, you need these huge screening funnels in order to narrow down.
So it's really the discovery phase then, the extra time is. And can you comment, Ben and Hugh, from pharma companies, based on the Inflation Reduction Act and the amount of time we're talking about to get a small molecule therapeutic in the first place, are your portfolios becoming more and more skewed away from small molecules? There is still a role for them, but are they diverging away from small molecules at all?
I mean, we've seen it in the charts earlier, right? The growth of the biologics, and this is in part response to the Inflation Reduction Act, and I think we're seeing this across the industry, right? People are starting to move away and try and push forward those larger biologics to avoid the complications of the Inflation Reduction Act. The other thing we are doing to try and overcome those challenges is speed up. How can we actually speed up this small molecule process? Because if we can speed it up, then we actually don't have a problem anymore. And that long time just adds cost. So how can we shrink everything? Using our traditional approaches, we've sped it up and shrunk it as much as possible. So now we're starting to look at other approaches like DNA-encoded libraries, to look at more chemical space in less time. That has its own challenges with deconvolution and pulling out the actual correct chemical entities. So there are a lot of things we can do to speed up small molecule drug discovery, and if we can achieve that, we could see the pendulum swing the other way again.
Okay, thank you.
Just do one more.
Okay. Hi, Michael O'Kane from Cool Reach Logistics. We've heard words like complexity, instability, novel movement of your material. When it comes to logistics, are there any parts of that space that can seek to speed up as well to ensure that we're getting the stability data that you need for the types of complexities of these shipments? Could the panel give their thoughts?
Davide, I don't know if you want to.
I cannot comment on other modalities, but what I'm very, very fascinated by is the idea to scale down. So with mRNA, you have the unique possibility to scale down production while retain cost effectiveness. So obviously, mRNA was first introduced for a pandemic, so we went for the global distribution chain. But there is the chance to scale down the manufacturing so that you can have distributed, even point-of-care manufacturing. And that would be very, very interesting to see how this unfolds. Certainly for supply chain, but also because allows to manufacture for batches where the population is equal to 1. So personalized medicines has this problem that are addressing very small population, ideally equal to 1, and then the cost effectiveness is not there. If you can scale down manufacturing, bring close to the patient without losing cost competitiveness, that would be great.
I think maybe just to comment on, I guess, and I think one of the panelists mentioned this, I think right now cell therapy is extremely concentrated in tertiary centers. I think we'll see over the coming years a significant widening of patient population, and therefore these tertiary centers are going to struggle to treat those patients. And I think how do you get supply chain to support regional hospitals to be able to cater for, cryopreservation on site, thawing on site, and be able to have the confidence for pharma companies or biotechs to have the confidence to get the drug in and back again. I think that's going to be a challenge over the coming years, and I know there are some good examples. UPenn, for example, in the US, have done a really nice job of a hub and spoke model, working with suppliers to reinforce the supply chain. So I think that's an area that we're starting to look at as well, because our therapy will potentially be an outpatient setting administration.
Great. Right.
Thank the panel, Hugh, great moderating, and for the panel for your insights. We'll go with that.